Okay, guys, today is Unit 5, Meiosis and Mendelian Genetics. Let's go over meiosis first. We mentioned meiosis on the previous unit review, but today, since I'm going to focus about maybe 10 minutes on it, we're going to go over it again. Meiosis is the division of a somatic diploid cell, or a diploid somatic cell, same thing, into four unidentical haploid daughter cells. Now, do any of you recall what type of cell this somatic diploid cell is that goes through meiosis? Sebastian? This is a germline cell. And what are the gametes called? These unidentical haploid daughter cells. What are the two types of gametes? Sperm and egg. So basically it looks like this. Here's our germline cell. It is diploid. It goes through one round of meiosis. This is known as meiosis 1. And it forms two haploid daughter cells. It then goes through a second round of meiosis, which is called meiosis 2. And these are going to result in sperm or egg. The whole point of this is to increase variation. If you are a sexually reproducing organism, you are entitled to a tremendously greater amount of diversity than asexual. Asexual is basically mitosis, which is like making a clone. The only way that they can really achieve diversity is mutations. Mutations are rare and they're random. But when you are sexually reproducing, you have tremendous amount of variation in every generation. Now, there's a couple terms that are associated with meiosis, and they are crossing over, a synapsis, and bivalent. Now, rather than draw this out, because it would take way too much time, I found a website that just has some images that I can show you and diagram for you. But there is another term I want to explain before I switch over to that image, and that is called homologous chromosomes. Homologous chromosomes are chromosomes that are paired because of common features. These features can be size, banding pattern, traits, and others. So let's say that this is chromosome number 1 that you got from your dad. So I'll color it blue for, I'll color it blue for dad. Let's try that again. So yeah, here's chromosome number 1. There's a band there, and a band there. And we'll color it blue. This is chromosome number 1 from dad. Here's chromosome number 1 from mom. This would make a homologous pair. This would be pair number 1. And for humans, how many pairs do we have, guys? Okay, I keep getting those numbers. When I ask for pairs, someone gives me the individual number. When I ask for numbers, somebody gives me the pair number. This has been a problem in this class, not second period, but this year, all year. You have 23 pairs. You have 46 individuals. Got it? Humans have 23 pairs of chromosomes. That means we have 46 individual chromosomes. Our diploid number is 46. Our haploid number is 23. So I'll ask you again, how many pairs of chromosomes do humans have? 23. This is the first pair. The reason why these two are paired up like this is because they have similar features. They are called homologous chromosomes. That's going to be important for you to understand before I get into the stages of meiosis, which I will get to right now. So I'm going to narrate this for you. There we go. Okay. I want you to check out interphase on the far left. Interphase in meiosis is the same as interphase in mitosis. You start up with G1, S, and G2. In G1, the organelles double, the cell doubles in size. In S phase, the DNA replicates. And then in G2, the cell is making the final preparations for division. But meiosis is different in mitosis, and the first aspect is that it has 2 rounds of division. Mitosis only has 1 round of division. In mitosis, you go from 1 parent cell to 2 daughter cells. In meiosis, you go from 1 to 2, then 2 to 4. The first round is called meiosis 1. I want you to look at prophase 1. The big difference between mitosis and meiosis when it comes to prophase is that in mitosis, you have the chromosomes just on top of each other to form one single file line on the metaphase plate. If you look closely at prophase 1, you have the homologous chromosomes have doubled themselves in S phase, and now you have sister chromatids that are homologous. And they go through something called crossing over, where pieces of the paternal chromosomes will exchange information with the pieces of, or parts of the maternal chromosomes. So you have a combination of both parents. You have dad's chromosomes, mom's chromosomes, they kind of get all meshed together, and they rearrange to form like a brand new chromosome. This is going to increase variation in this individual's offspring. You then go over to metaphase 1, and you can see this is the forming of a bivalent. A bivalent is where the homologous chromosomes are side by side with each other, and they've already gone through crossing over. So you have little pieces of the maternal chromosome on the paternal and a little piece of the maternal on the paternal. This just increases variation. In anaphase 1, the sister chromatids get pulled apart. And then in telophase 1, you're going to go through the reforming of the nucleus around the DNA, and you're going to have 2 daughter cells. But then we get into meiosis 2, where it happens all over again. Now, if you were asked what stage of meiosis is more like mitosis, I want you guys to look at the 2 of them. Look at meiosis 1 and look at meiosis 2. Which one looks to be more like mitosis based on what you know? Meiosis 2 is more similar to mitosis than meiosis 1 is. So if you follow the stages of prophase 2, metaphase 2, anaphase 2, telophase 2, it looks very similar to mitosis. You ultimately end up with how many chromosomes are in each of those daughter cells? Okay, so does that tell me that with this being meiosis, is the haploid number 2 or is the diploid number 2? The haploid number is 2, so what must the diploid number be? The diploid number must be 4. If you look there, there's the original cell. How many chromosomes are in that? So the diploid number is 4. The haploid number is 2. Got it? Sure? If you need to slow this video down, rewind it, or if you even go to Google and you type in stages of meiosis, you can find great resources and go at your own pace. All right, let's go back here. And I want to talk about when meiosis goes wrong. We have euploidy, and we have aneuploidy. Euploidy is the normal number of chromosomes. Humans, this would be 46. Dogs, this would be 78. Aneuploidy is an abnormal number of chromosomes. If you have Down syndrome, you have 47 total chromosomes. My aunt had 45. She was missing one of her X's. She was aneuploidic. This all happens because of non-disjunction. Non-disjunction is an error in meiosis when homologous chromosomes fail to divide. So I would love to draw it out for you and just take it piece by piece, but I have an image here to show you, another thing from online. So let's take a look at that. I want you guys to take a look at, this is right here on the far left, that is normal. I want to see if you can tell me. Look at the before box. What is the diploid number? 4. Okay, it is 4. And then what is the haploid number on the bottom? 2. Very good. Now take a look at when non-disjunction takes place in meiosis 1. How many chromosomes should there be after meiosis in each cell after meiosis 1? How many are there in the daughter cells after meiosis 1? 3 and 1. So if you notice in meiosis 1, here's what I want to ask you. What are the chances that a cell will be aneuploidic if non-disjunction takes place in meiosis 1? 100%. You have 50% trisomy, which is N+1, and you have 50% monosomy, which is N-1. Now keep in mind that this will not be every single chromosome pair. It's just one pair. People that have Down syndrome, they have 2 chromosomes in pair 1, they have 2 chromosomes in pair number 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. But then you get to the 21st pair, there's 3. And then their 22nd pair is normal, and their 23rd pair, the sex pair, is normal. So they're called trisomy 21. Take a look at when non-disjunction takes place in meiosis 2. What are the chances that a cell will, or an individual will be aneuploidic? 50%. So if you had to choose, you would want to go with meiosis 2 because you still have a chance of having some normalcy. If non-disjunction takes place in meiosis 2, you have a 50% chance of the cells being normal. You have a 25% chance of having trisomy. You have a 25% chance of being monosomy, which 25 and 25 is 50% of being aneuploidic. All right? All right, so that's basically it for meiosis. Let's jump on over to Mendelian genetics. What is Mendelian? Who is Mendel? What's his first name? You remember? Gregor Mendel. He was an Austrian monk, and he was fascinated with pea plants and science. And he worked with his little garden, and he had flowers and peas, and he tried all these different crosses to have different outcomes. And these were called reciprocal crosses. So what he did is, remember, this is all a summary. He had a tall pea plant, and he crossed it with a short pea plant. That's the pea generation, the original generation. The generation that comes from that is known as F1. Do you recall what the phenotype is of all the plants that came out in F1? No, Sebastian. Kylie? They were all heterozygous, so what was their phenotype? They were all tall. So he thought, okay, he made hundreds of these. He crossed a tall plant with a short plant, and they all came out tall. Remember that the prevailing thought in the day was the blending concept of inheritance. So you have a tall parent and a short parent, you're going to have intermediate offspring. Well, this contradicted that. Here we have a tall parent and a short parent. You have a tall offspring. Do you recall what he did next? He crossed F1 with each other. But before I get to that, I want to be sure you guys understand. He did something called a reciprocal cross. A reciprocal cross is where he took pollen from the tall and put it into the female flower parts of the short. And then he did vice versa. He put pollen from the short into the female parts of the tall. So that is known as a reciprocal cross. But in the F1 generation, he let the tall and the tall from F1 cross with themselves. So basically, you're crossing one with it in and of itself. So they're self-pollinators. It just pollinated itself. Self and self. And do you remember what the outcome was for F2? What? It was a 3-to-1 ratio. For every 3 tall, there was 1 short. And so what Gregor Mendel realized is that the gene skipped a generation. It didn't go away. It was covered up. It was masked. This led to several ideas, and I'm going to write them all down here. The first one is we have genotypes. This is the genetic composition of a trait. Oop. Sorry about that if you couldn't see. So I didn't share that one, so I'll just show you guys, I'll let you take that in. Again, the P generation, he crossed tall with short, and he got all tall. So he thought, what happened to the short trait? It's gone. He let the tall cross with itself, and we got 3-to-1 ratio. For every 3 tall, there was 1 short. So the first term I'm going over here is genotypes. This is the genetic composition of a trait. There's actually 3 main genotypes, but there's either homozygous, where you have the same alleles, and I'll get to alleles momentarily. Now, do you remember what is another word for homozygous? It's actually 2 words. It's hyphenated. True breeding. Another word for homozygous is true breeding. So we could say that one parent was true breeding tall, the other parent was true breeding short. If you're homozygous dominant, then you have all dominant alleles. If you're homozygous recessive, then you have all recessive alleles. All right. Then we have phenotype. Phenotype is just the physical expression of a genotype. So when I'm saying tall and short, those are phenotypes. And when I say homozygous or heterozygous, that would be genotypes. All right. Another term, and actually I'll get to what I was thinking momentarily, but we have alleles. These are just forms of a trait. So in this example, we have the dominant allele, which is tall, and we have the recessive allele, which is short. So it turns out in Gregor Mendel's experiment, if I can go back up here, this first parent was homozygous dominant. We could also call it true breeding tall. This short parent was homozygous recessive. We could call it true breeding short. It produced all heterozygotes. Can we use the word true breeding with a heterozygote? Nope. It's a hybrid. The opposite of true breeding is a hybrid. So this was a hybrid, and when you cross the hybrid with a hybrid, the expected outcome is 3-to-1. 3 being tall, 1 being short. Now this leads me to a couple other terms I want to be sure that we get, and that is called complete dominant versus incomplete dominant. Okay, let's see if you guys can figure this out. You ready? Let's say that P height is a dominant trait. Here's my genotype, that is homozygous dominant. What would the phenotype be? Here's heterozygous, that is the genotype. What would the phenotype be? And here's homozygous recessive, that is the genotype. What would the phenotype be? Short. If a trait is completely dominant, that means that the dominant allele overwhelms and overrides the recessive version. But let's go to incomplete dominant. Let's just pretend that P height in P's is an incomplete trait. Here's the homozygous dominant genotype. What is the phenotype? How about heterozygous? Medium. And how about homozygous recessive? Short. So whenever you have an incompletely dominant trait, it's a blend of the two. It's an intermediate of the two. So tall and short would be medium. Red and white would be pink. Okay? Let's get to codominance and multiple alleles. I can explain both of these with one example. Codominance is when two traits, or excuse me, two versions of traits are expressed equally. And multiple alleles is just where you have more than two versions of a trait. All right, so the example that we can use for both of these is the A, B, O blood groups. This is the B allele. This is the A allele. This is the O allele. The fact that there are three alleles, three different alleles there is an example of multiple alleles. Again, these are the alleles. When it comes to the genotypes, if you had type B blood, you would either have homozygous dominant or heterozygous. If you had the A allele, or excuse me, the A genotype, you would either be homozygous dominant or heterozygous. If you were O, you can only be homozygous recessive. If you're AB, that would be heterozygous between A and B. This one right here is codominant. If you have B blood type, that means that your cells are covered with B antigens. This is your body's way of identifying that this is your cells. Anything that is not B is not you. If you have type A blood, this means that all of your cells have the A antigen, and anything that's not A is not you. If you have type O blood, which is the rarest, this means you have no antigens, so anything that has antigens is not you. And the rarest blood type in the world is AB. You have A antigens and you have B antigens. It's equally expressed. That is an example of codominance. Here's something else with multiple alleles that we did. If you remember this with the bunny rabbits, this was a black rabbit. This was a chinchilla, which is like the color gray. This is Himalayan, which is white with the black spot. And this was white. I want to throw a few genotypes at you guys. Why don't you tell me what the phenotype would be, okay? Here we go. I'll do like 4. Tell me the color. This is a big C, by the way. What's the color? Black. It's in order. The top is the most dominant. The bottom is the least dominant. How about this? No. Where do you see the chinchilla would be this? That's Himalayan. What is this? What is the only genotype that could be white? So, what's that an example of? All those different alleles. That's an example of multiple alleles. All right, let's see what else we have here. All right, law of independent assortment. Again, there's a lot to go over. Trying to squeeze all this in. The law of independent assortment states traits are independently passed on. The fact that Sebastian has dark hair has absolutely nothing to do with the fact that he had crooked teeth as a kid. They're totally different traits. When I had a big, goofy from Disney World gap in my teeth growing up, I needed braces really bad. That has zero relationship with the fact that I'm tall or that I have blue eyes. Every trait is independent of another trait. Every trait's independent. The other law is the law of segregation. This means that homologous chromosomes separate during gamete formation, which is meiosis. Okay, but there is an exception, kind of an exception. It's called linked genes. Now, these are genes that are not related to each other, but the fact that they're so close to each other on a chromosome means that they're likely going to be passed on together. So this is just close proximity to genes on a chromosome insinuates they are inherited together. So here's a chromosome. Let's say here's gene A, B, C, and D. Which two genes do you think are going to be most likely passed on together? Which ones, let me say, have the greatest likelihood of being passed on together? B and C. Why? Because they are so close to each other. We could say that there's maybe like a 25% chance that A and B will be passed on together. There's maybe a 25% chance that C and D will be passed on together, but for B and C, it could be like 90% chance. Now, it doesn't all have to add up to 100. But because B and C are so close, they're likely going to get passed on. That's an example of linked genes. Okay, let's see what else we have here. Let's do a monohybrid cross. A monohybrid cross is when you do your Punnett squares. What frequency does each box represent? 0.25, 25%. So let's say that I was crossing these two parents. Homozygous recessive crossed with heterozygous. Let's do that. What are the odds that you would get a heterozygous offspring? 50% in a homozygous recessive. So what this does, everybody, is this provides expected outcomes. And in this particular example, we would expect 0.5 tall and 0.5 short. What math equation did we learn this year that has to do with expected and observed? Chi-squared. It's pronounced chi-squared. So I want to use this example of monohybrid crosses to teach you, to review with you chi-squared. So let's say that I do, here's my problem. I cross a heterozygous plant with a homozygous recessive plant. Again, I'm trying to save time, so I'm abbreviating my words here. 10 seeds are formed and result in 7 tall and 3 short. Is this likely? So what you need to do is you need to do the whole chi-squared equation. So chi-squared is the summation of observed minus expected squared over expected. So let's do the tall individuals first. Observed, how many did I have? 7. Expected, how many would I get? Well, you have to ask yourself, what was the total number? What was the total number? 10. And look at the Punnett square, guys. There's a reason you were given this and this. You have to do the Punnett square. And according to the Punnett square, what would be the expected frequency of having tall offspring? 50%. And 50% of 10 is 5. Then you're going to add that with, what was the observed for short? What was the expected for short? Okay, you will have a calculator on your exam, so give me a moment while I figure this out. 7, why did I put times? Excuse me, it's minus. I don't know why I did that. 7 minus 5 is 2 squared equals 4. Divided by 5 is 0.8. And then we have 3 minus 5 is -2. -2 squared is 4. 4 divided by 5 is 0.8. So my chi-squared value is 1.6. That's my chi-squared. Everybody so far so good? You would then need to look at your formula sheet, which I have up here on the ready. Let's go back up here. There you are. Okay. Which confidence value do you typically look at? 0.05 or 0.01? 0.05. It's called your p-value. And how many degrees of freedom were there? We have tall and we have short. So how many degrees of freedom is that? 1 degree of freedom. So you look where 0.05 intersects with 1 degree of freedom. What is our critical value? 3.84 is correct. So our critical value is 3.84. So what value is greater, the critical value or our chi-squared calculated value here? So whenever chi-squared is less than your critical value, what do you do with your null hypothesis? Do you accept it or do you reject it? You accept the null. And accepting the null generally means it is possible to have 7 tall and 3 short based on the parent's genotype. There's nothing wrong with that. It's completely likely. If you were to reject the null, then you would say, no, that's not possible. Something's wrong. Something's going on. There's some outside influence that's making this happen. There is a link here. The null is saying there's no issues here. There's no conspiracies. Everything's normal. All right, let's do a test cross. Let's say in Labrador retrievers. Let's just pretend, I'm going to make something up. This is not true. Let's say that if you are a black lab, that's dominant. If you're a yellow lab, that is recessive. I have a yellow dog that I want to cross with a black dog. I don't know for sure what my black dog's genotype is. Do I know for sure what my yellow lab's genotype is? What is it? Homozygous recessive. That's the only possibility. What could my black dog be? How many? It has at least how many alleles? Or dominant alleles? It has at least one dominant. I don't know what the other one is. You follow so far? So what we need to do is we need to cross the 2 of these to find out whether my black lab is dominant or heterozygous. And so I cross them, and they have puppies. They have 8 puppies. 2 of the puppies are yellow. What does that say about my black lab? He's heterozygous. The only way that a parent with a dominant trait could produce offspring that has a recessive trait is that the parent with the dominant phenotype is a carrier. This is known as a test cross. A test cross is where you cross an individual with a recessive trait with an individual with a dominant trait to determine if the individual with a dominant trait is homozygous dominant or heterozygous. Let me fix my computer real quick so it's not getting power. Here's why. There we go. All right. So if anybody's seen me, I have brown hair. In humans, is brown dominant or recessive? If I have a blonde child, what does that say about me? So the whole point of doing a test cross is you can see that an individual is showing the dominant trait, but you don't know for sure what's going on in their genes. So you're going to test it. If I produced a blonde child, or if I help produce a blonde child, that means I'm heterozygous. Because the only way that blonde child can get that blonde hair is if the mother and the father provided the recessive allele. Okay, let's keep it going. Let's do a dihybrid cross. Now, if you guys do a dihybrid cross, it looks like this, you're torturing yourself. If you do this during the AP exam, you're nuts. Let's say that you're asked to find out the frequencies of a trait like this. What can you do instead of doing this crazy dihybrid cross? Do 2 monohybrid crosses. Okay, what are the chances that an individual will have a dominant phenotype on the G's? 0.5. And how about recessive? 0.5. How about over here for the H's? It's also 0.5 and 0.5. That worked out great. So you just have to multiply the frequencies. If I want to know, guys, if an individual is going to be dominant, what's 0.5 times 0.5? 0.25. What if they have the dominant slash recessive? You would do 0.5 times 0.5 again. A cross like this would be a 1-to-1-to-1-to-1 ratio. Do you remember what it is if you have heterozygous cross with heterozygous? 9-to-3-to-3-to-1. If you want to know what frequency that is, you just do 9 divided by 16, 3 divided by 16, 3 divided by 16, and 1 divided by 16. I've been doing this so long, I've memorized the numbers. This is 0.5625. This is 0.1875, 0.1875, and 0.0625. So in order to get an individual that's homozygous recessive for both traits, that's 1 out of 16. You would multiply the frequency times 0.0625. So if I have a population of like 500 bugs, I want to know what percentage of them are going to show the recessive trait for both. You would do 500 times 0.0625. And that would tell me, let me do my math, 500 times 0.0625. Out of 500 insects, I would expect, keyword, expect 31.25 to be recessive, to show the recessive trait for both. That's something that a lot of people were getting stuck with, is when I ask them, how do you find out how many individuals have this trait or that trait? They didn't know. You multiply the total times the frequency. Okay? You multiply the total times the frequency. You might get a chi-square problem like that. You might be asked, how many individuals are going to be dominant? We had a problem like that in the mock exam. Remember that you need to get the total. And this 9 to 3 to 3 to 1 is out of 16. If you look at the Punnett square, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. That's where the 16 comes from. Okay. All right, we have about 9 minutes left. Let's see if I'm missing anything. I'm looking at my long laundry list of stuff here. Let's do pedigrees. Remember that circles represent females, squares represent males. I'm going to draw a pedigree here real quick. I'm going to try and not make this too long. I know it's sloppy, guys. I'm just trying to get through this as fast as I can. Okay. How many generations are on this pedigree? How many? 3 generations is correct. What do we call the first generation? P. What's the second one? F1. What's the third one? F2. It just goes on after that. How many people in this pedigree are affected by the trait? 2 people. Now, did this trait skip generations? Yes. Did this trait have parents that, let me rephrase that. Is this trait found in people that had unaffected parents? So does this tell you that this trait is a dominant form or a recessive form of the trait? This tells you it is recessive. But now we need to figure out, is it autosomal or sex-linked? Autosomal means gender doesn't matter, it's a body chromosome. Sex-linked means gender does matter, it's on a sex chromosome. So do we have males only affected or both genders? Okay, can females have sex-linked traits? Yeah, but what has to happen for a female to have a sex-linked trait? Her father has to be affected too. So take a look at our female right here. Does she have an affected father? So is it sex-linked? Nope, it is an autosomal recessive trait. Okay, so there's 2 ways to tell if it's dominant or recessive. If it is a dominant trait, it's not going to skip generations. If you have parents that are affected, there's like a 50-50 chance that their offspring could be affected. You can tell that it's recessive if it skips generations, and if unaffected parents produce affected offspring. That's the one that people have been forgetting. Unaffected parents produce affected offspring. That's a sign that it is recessive. Let's get to sex-linked traits, everybody. These are traits carried on what chromosome? Carried on a sex chromosome. You can have X-linked, you can have Y-linked. Talking about colorblindness. I'm a student who's colorblind. A male student. His father is not colorblind, his mother is not colorblind. Who did he inherit the trait from? His mom. Here's what he looks like. That is his genotype. He tells me his father's not colorblind. So what type of allele is on his father's X? A dominant or a recessive? And there's the father. What chromosome did this boy, this male student of mine, inherit from his father? His father's X or his father's Y? That's right. Now he tells me that his mother is not colorblind either. So What type of allele is it on at least one of mom's X's? But what about the other one? So he inherited this one from his mom, right? Can females be a carrier for an X-linked trait? Can males be a carrier for an X-linked trait? No. So let's go over all the possible genotypes. This is homozygous dominant. This is heterozygous. This is homozygous recessive. The only way for a female to get a sex-linked trait is that her mother has to be a carrier and her father has to have the trait. Because all you two girls in here, which chromosome did you inherit from your father? His X or his Y? You got his X, and your other X came from mom. Here is males. We call this hemizygous dominant, and this is hemizygous recessive. Males cannot be a carrier for a sex-linked trait because males only have one X chromosome. Okay. Let's see. Let's talk about a karyotype. I don't have time to draw the whole karyotype. Do you guys remember how it looks where you have like pair number 1, pair number 2? Remember that? Pair number 3. What if I go down here to 21 and there's 3? What would that be indicating? Trisomy 21. What's another name for that? That's Down syndrome. Do you remember what this is? Here's the sex pair. What gender would that be? That's a male because what's bigger, the X or the Y? Yeah, so this would be a male. This would be a female. Follow? The X chromosomes are pretty big. The Y chromosome is very, very small. What if we had a karyotype to look like this? Do you remember what the name of this is called? You need to know this. 2 X's and a Y. Now this is trisomy, but what's the name? It's called Klinefelter. You need to know that one. There's 2 sex-linked traits, or there's 2 aneuploidic sex traits you need to know. Klinefelters were your XXY. Now, what is this one? What if you just have one X? What is that called? Turners. Those are my antads. Okay. Guys, I think that's about it. That's all the time I have for this. So there is some more stuff. I would recommend you go back on YouTube and look over some of my lessons from Unit 5. But all in all, that's about it.